Welcome, Honors Biology. Today I'm going to go over Chapter 12, which is the chromosomal basis of inheritance. As I've mentioned in class and in other videos, we're continuing to build on this idea of genetics and chromosomes and genes, and since we've done things a little bit out of order, I think you'll find that Chapter 12 actually pulls a lot of disparate ideas together in a way that you're already prepared to understand. We just haven't explicitly defined some of the things that are going on. So what I think you'll find is that Chapter 12 is really building on Chapter 11, adding a few more skills, but most of the things you already understand from Chapter 11 will still work for you here in Chapter 12. Since it's on the chromosomal basis of inheritance, we begin by looking at a chromosome. So remember, chromosomes were those structures that we can see inside the nucleus, and in this particular chromosome, there's a portion that's a different color, fluorescently tagged, indicating a gene on that chromosome. So we already know this because we've gone through the molecular basis of DNA, but all of Mendel's hereditary factors, what he described as hereditary factors, were genes. And we know those genes are located on chromosomes. And so when we move from Mendelian to chromosomal theory of inheritance, we basically say that Mendelian genes have a specific loci or a position on a chromosome, and these chromosomes undergo segregation and independent assortment through meiosis. So we're really fusing what Mendel saw with what we learned about meiosis to come up with this chromosomal theory of inheritance and really put the laws of segregation, laws of independent assortment into some sort of context to try to understand what's going on inside of our cells. What this does is it makes some inheritance patterns that are a little more complex and creates some more diversity in offspring of parents, which is pretty important for evolution. So as you can see here, we see a simple dihybrid cross, just like we looked at in the last chapter. So green wrinkled and yellow round seeds, we see them going through first generation to make an F1 generation that is heterozygous for both alleles, big Y, big R, little r. And then we allow that to go through meiosis again to get us the 4 possible gametes, which when combined with itself to give us an F1 cross, and F2 gives us the 9-3-3-1 set of phenotypic ratios that we expect. I talked through this really quickly. This chapter is not actually about this topic. This is all from Chapter 11, but hopefully this looks familiar to you. But all we're doing is we're adding in this idea that the reason these ratios arise are because of the way the chromosomes separate during meiosis. I don't really distinguish between those two ways of thinking when we teach it, so you've seen this before, but just as a frame of reference, let's make sure we're putting meiosis into the context of the genetics that we're thinking about and recognizing that meiosis is what ultimately causes us to get the ratios that we expect to see. Alright, so where did it get a little more complicated is in the early 1900s. Thomas Hunt Morgan began to study fruit fly, Drosophila melanogaster, instead of looking at things like plants. So fruit flies are a convenient organism for genetic studies. They're still used by many scientists around the world because fruit flies produce many offspring. They can be bred every 2 weeks, and they only have 4 pairs of chromosomes. So it's a lot easier to figure out what's going on with 4 chromosomes instead of 23 pairs like we have in humans, and you can make a lot of flies really fast, as you've probably experienced if you've ever left some fruit sitting out on your counter or anything like that. So Morgan was actually, Morgan and folks in his lab, had flies around. They were collecting flies. It's pretty easy to get fruit flies from the wild if you, like I said, if you put out some fruit or some vinegar. And they noted that there was a large population of what they called wild type or normal phenotypes, that basically if you pick up a fruit fly and if it looks exactly like what you expect a fruit fly to look like, that's a wild type fruit fly. And then they started identifying alternative traits, things that were different from the wild type, and we call those mutant phenotypes. So Morgan started distinguishing between wild types and mutant phenotypes in flies, and the first phenotype he discovered was a fly with white eyes instead of a wild type red-eyed fly. So if you were to find a Drosophila fly in the wild, it has red eyes, but here's a mutant that instead of having red eyes, it has white eyes. So as you do when you're studying genetics, he would start mating these flies together. So he took male flies that had white eyes with female flies that had red eyes, And he got F1 generation that had all red eyes, and an F2 generation that had a classic 3:1 red to white ratio, which you would expect if white is recessive to red, assuming that the male is homozygous recessive and the female is homozygous dominant. You would get all heterozygous in the F1 generation, and the F2 generation would be 3:1, your classical Mendelian ratio. But what Morgan noticed was that only the male flies had white eyes in this scenario. So female flies all had red eyes, only the males had white eyes, which is kind of peculiar. So Morgan concluded that the eye color was actually related to the sex of the fly, and I'll go ahead and give you a term for this that I'll show you what it looks like in a second. The male flies are actually hemizygous. They only have one copy of the allele. So they're not homozygous, they're not heterozygous, they're hemizygous. They only have one allele. That's kind of weird to wrap your head around, so let's look at a picture of it. So here's the experiment. So a red male, a red female, a white male. All the offspring have red eyes, and then you get 3:1, but all of the males, all of the white eyes are male. And so the way that Morgan made sense of this is that he had to actually put the allele for white eyes on the X chromosome. So if you are a female fly, you have 2 copies of the X chromosome. If you are a male fly, you only have 1 copy of the X chromosome. So having 1 recessive copy makes you hemizygous for white. You have white eyes, but there is no way, if you combine this white eye with this red-eyed wild-type female, the males are now wild-type, the females are now heterozygous for white. If you combine the heterozygous with a male that is hemizygous dominant, the only way you can get a recessive phenotype is in the males. Females can be carriers, but they cannot actually have the trait. You'll have to play with this. We'll go over this in class, but make sure you think about what's happening here is that when we put it on the X chromosome, or in some cases a Y chromosome, the pattern just becomes a little bit different, even though the way we would set up a Punnett square is fundamentally the same. So that leads us, Morgan's discoveries lead us to thinking about sex-linked genes. And so that's one of the big topics of this chapter, is that there are sex-linked genes that exhibit unique patterns of inheritance because they are on those sex chromosomes. So humans and other mammals have 2 types of sex chromosomes. I'm sure we've talked about this before, an X chromosome and a Y chromosome. Y and X are homologous in small portions, but they're generally not homologous. Females are XX, so females have 2 copies of X, males are XY. So males have 1 X and 1 Y. Just to give you a caveat on this, other animals have different sex determination methods, different types of chromosomes arrangements, but we'll generally talk about humans because that's the easy one for us to think about XX and XY, and flies are the same. You can see, and this is always something that's funny every year for boys to recognize, is that the Y chromosome is way smaller than the X chromosome. So the Y chromosome pretty much only contains genes for making males male. The X chromosome contains all sorts of things that are important for normal physiological function. So everyone has at least 1 X chromosome. Only males have a Y chromosome. You can see this in zygotes, how this works. So male sperm can have either an X or a Y because males are XY. All female eggs are X. So when you combine them, it is the sperm from the man that determines ultimately whether the offspring are male or female, because an XX will be a female, as we've said, and an XY will be a male. So any gene that's located on one of these sex chromosomes is called a sex-linked gene. There are a few Y-linked genes, not very many, we don't tend to talk about them, and there are a good number of so-called X-linked genes, genes that are on the X chromosome. X chromosome genes are lots of characteristics unrelated to sex, and if you have a gene that's on the X chromosome, a female needs to have 2 copies of that allele. She needs to be homozygous recessive. That's the only way a female can have that trait, but a male only needs 1 bad copy of the allele, or 1 mutant copy of the allele, because he is hemizygous. Males only get 1 X, so whatever's on that X is what they're going to express. Females get 2 X, so dominant and recessive relationships still matter. That makes X-linked recessive disorders much, much more common in males than in females, although it doesn't make them impossible to get in females, as you'll see. So here's an example, and one thing you'll see here, similar to the way we did blood type and the way we did some of the codominance traits, we'll put X with a superscript for the allele. So here's an X with a capital N, X with a capital N, so that makes this female homozygous dominant for whatever the N trait is. The male is X little n y. That makes the male hemizygous for whatever the recessive trait is. So the male is going to express the recessive trait. The female is going to be homozygous dominant. When you set up this Punnett square, the first thing I recommend is set up the x's and the y's. So eggs are going to be XX, sperms are going to be XY. Eggs will have a capital N or a capital N. Sperm will have a lowercase n or there'll be a y. And then you can make this Punnett square where you'll see this half is female, XX, this half is male, XY. In this particular arrangement, these males are all hemizygous dominant. They all have the dominant trait. The females are all heterozygous now. They all have one copy of the dominant trait and one copy of the recessive trait. If you were to take these offspring and mate them again, so here's a heterozygous female with a hemizygous dominant male, you see that the females are all going to have the dominant trait, although half of the females will be homozygous dominant and half of the females will be heterozygous. The males will be half and half. Half will have the dominant trait, half will have the recessive trait. You'll have to get a piece of paper and play around with these, and I think they'll make sense for you. Sometimes students wonder, how could a female ever get one of these X-linked traits be recessive? And that would happen if the mother was a carrier. So here's a heterozygous mother, a hemizygous recessive father. That means that of the girls, half will be carriers, heterozygotes, and half will be homozygous recessive. Of the boys that this cross generates, half will be dominant and half will be recessive. So again, Punnett squares will still work the same way Punnett squares will work. You'll still do probabilities the way that you've done probabilities, but we now add this wrinkle of thinking about the X and Y chromosomes and recognizing that we now have kind of a second thing going on, whether you're male or female, that will impact what sets of chromosomes you get. So some of these recessive X chromosome alleles that are common, that you'll see commonly in questions. Color blindness is mostly X-linked, so men tend to be colorblind more than females. Duchenne muscular dystrophy and hemophilia are also X-linked traits, so you tend to see these occurring more in men than in women. So another interesting feature in women, because women have 2 X chromosomes, in female mammals, only one of the 2 X chromosomes is activated, and the other is inactivated randomly during embryonic development. That means that even though we say every cell has the same set of chromosomes, in women, one of their X chromosomes is inactivated and condenses into something called a bar body. So any female who's heterozygous for a particular gene on that X chromosome will actually be a mosaic throughout the body for that characteristic. It's kind of weird to think about, but I think the classic example that will make sense to you is if you think of a calico cat. So on these kinds of cats, the color, the fur coloration for orange or black is found on the X chromosome. So if a female cat is heterozygous for orange and black, in some cells the orange chromosome will get deactivated, in some the black will be deactivated, and that's how you get a calico female who has orange and black patterns all over her body. You would not get a calico male with this particular set of chromosomes and genes, because the male is either going to have a black X or an orange X. It'll be one color or the other. You will not be able to get a so-called calico male in this format. So the last idea about chromosomes to think about is that, aside from the XY adding some complications, there are some genes that are inherited together because they're located near each other on the same chromosome. So everything Mendel did, and I told you about this in the last chapter, he sort of selected because he got nice clean arrangements of offspring that really supported his laws of segregation and independent assortment. As it turns out, when Morgan started looking closer and when other scientists started looking closer, there are some genes that don't have those nice clean breaks between offspring, and genes that are on the same chromosomes that inherit together are called linked genes. So we're going to add this concept now of linked genes as we think about genetics. So Morgan was doing these experiments with fruit flies, and if you look at two traits like body color and wing size, he saw that they were usually inherited together in specific combinations. So you would either see big and black and big wings, or you'd see gray and small wings. He reasoned that since these genes did not assort independently, that they were on the same chromosome. So if you think about that model of meiosis, how we use meiosis to recognize how genes are independently assorting, if you put two genes on the same chromosome, there's really no way for them to separate apart from each other. So you're going to get two traits that are tracking with each other throughout all of the meiosis. And you see this in this picture. So here is a dihybrid female. So she is, for all intents and purposes here, fly people use plus signs instead of capital letters. So dominant for B, dominant for VG, recessive for B, recessive for VG. The male is recessive for everything. The offspring are either going to be heterozygous or they're going to be recessive, as you would expect, for both traits. So you're going to get either looking like the mom or looking like the dad. We would call these parental offspring. What they found, though, is that they got some non-parental phenotypes as well. So they got some phenotypes, instead of being B is for black, VG is for vestigial wing. So instead of having the normal color and the normal size wing, some would have the normal color and a small wing. Some would have a normal wing and a different color. They got what they called non-parental phenotypes. They didn't look like any of the parents, and the only way to understand this is through genetic recombination, that phenomenon we saw in meiosis where the chromosomes cross over during prophase 1 of meiosis 1. So here is your wild-type fly, gray body, normal wings, so B+, VG+. Here's your double mutant, black body, vestigial wings, B, VG. So when you cross these, you get a heterozygote. If you mate this heterozygote with a homozygous recessive, you would expect to get only the parental phenotypes. You should get 50-50 gray big wings and 50-50 black small wings. But what you see if you actually do this experiment is that if the genes are sorted independently, if B and VG are completely independent, you would expect to get a 1-to-1 to 1-to-1 ratio. If they're completely dependent, if they only sort together, you would only get a 50-50, like I just mentioned. But the results that you get from this kind of experiment is 965 to 944. Okay, so those are basically 1-to-1. But then you've got 206 and 185, which doesn't fit either of these two predicted ratios. So we don't see them being fully on the same chromosome. We don't see them being fully independently assorted, and so this begs the question, like, what is going on here? And so when we think about these parental types and their recombinant types, generating recombinant phenotypes, combining the traits, would be 50%. It would happen 50% of the time if you were on different chromosomes. So Mendel's models basically predicted that if you do a dihybrid cross, there's a 50-50 chance of getting either trait. Morgan's models, suggesting that they were completely linked together, would basically say you'd have a 0% chance of having a recombinant set of traits, a new combination, because they're so tightly linked together. Anything in between 0 and 50 is showing some kind of linkage, and we needed to figure out what that means. So here are your P's again that we would see in the last chapter. The yellow round and the green wrinkled are the parental types. The yellow wrinkled and the green round are the recombinant types. So you see we flip those phenotypes around. So I already kind of told you this, but how do we get this? We get this from crossing over. So remember we talked about crossing over being a fairly rare event, but it allows a chromosome to combine with its homologous chromosome during meiosis and basically mix and match some pieces and parts. The mechanism for creating these recombinant phenotypes is that crossing over phenomenon. So if we think we've got a gray with normal wings, we've got a black with small wings, and we create that F1 dihybrid, right? So B plus VG plus B VG. If we allow that to do some recombination, the eggs that we're going to get from this fly are going to have 4 different types of chromosomes. We're either going to get B plus VG plus, right? That's this one, or BVG, that's this one. That's going to happen most of the time. But with some frequency, you're going to mix and match between these two loci and get a B plus with a VG and a B with a VG plus. You're going to get these recombinant chromosomes. Now, something that's a little odd in flies, and we're not going to worry about it too much, is that male flies don't do recombination. This fly actually doesn't need to do any recombination anyways, because everything he has is the same. So even if these were to mix, you wouldn't see anything, but you know, the male is only getting one kind of sperm, but the offspring we get now have 4 different phenotypes. We have a wild type that's gray and normal. We have black with vestigial wings. We have gray with vestigial wings, and we have black with normal wings, but you'll notice that the recombinant offspring happen at a much lower frequency, 206 and 185, to the wild type and the black vestigial, the parental type offspring. So that shows you that these recombination events are fairly rare. Moving even beyond this rarity of this event, we can calculate the recombination frequency. It sounds like a complicated number, but the recombination frequency is simply the number of recombinants. So 391 is 206 plus 185 over the total offspring. So this total experiment gave us 2,300 offspring. So we got a recombination event 391 times out of 2,300 attempts, or 2,300 offspring. Multiply that by 100 to give us a 17% recombination frequency. So these two genes will recombine 17% of the time. And remember, I already gave you those boundaries, right? So if they're perfectly linked, they'll recombine 0% of the time. If they're completely unlinked, they'll recombine 50% of the time. So 17 sits somewhere between 0 and 50%. So the random combination, why is this important? It creates a lot of variation for natural selection. So these bring different alleles together in new combinations, and then we add on top of that random fertilization that we talked about in the last chapter, and this is how we get natural selection, all of this mixing and matching. So don't lose sight of the fact that all of these mechanisms are creating a lot of variation for us. Another benefit of kind of doing this recombination frequency calculation is that we can use recombination frequency as a measure of how far apart on a chromosome 2 particular genes are, how far the loci are. So the further apart the genes are, the higher the probability is that there's a crossover. The lower the number is, the less of a probability. So the lower it is, the closer they are. The higher the recombination frequency is, the further apart they are. Up until you get to 50% recombination frequency, which suggests they're either on separate chromosomes or they're so far apart on a particular chromosome that they basically cross over freely whenever it wants to. So before we had the genome, before we had like really molecular ways of understanding, scientists actually generated these linkage maps, genetic maps of chromosomes based on the recombination frequency, using map units that equated to 1% recombination. And by doing series of experiments and mating this with that and figuring out all the recombination rates, they were able to do pretty good mapping of a chromosome. So if you look at that black and vestigial wing that we looked at before, you know that they're 17% apart from each other, 17 map units apart. If you then compared black to the CN gene, you know that's 9%. VG and CN is 9.5%. You probably did different arrangements here, and you can kind of put them together into a puzzle and figure out how the chromosome is all laid out to ultimately give images like this that were the sort of first looks at how things were arranged on a chromosome and where specific genes exist on a chromosome. We've gotten much more precise with this now, and different organisms, recombination units, map units, recombination frequencies relate to different numbers of nucleotides. It becomes kind of a complex comparison there, and it's really a difference between geneticists and biochemists, where they kind of approach the problem different ways, but it all aligns with our understanding of genes being located on different places on chromosomes. So the last idea to talk about here, and I know chapter 12 is kind of tying up loose ends and giving you lots of ideas, but the last idea is that chromosome numbers or structures can be changed kind of wholesale in big ways that will also cause genetic disorders. So we've talked about how X and Y segregate differently, and that can create some variation. We've talked about how crossing over of chromosomes can create some variation. Now we're talking about large-scale alterations to chromosomes. So an entire chromosome missing, a portion of a chromosome missing. These can often lead to spontaneous abortions or cause pretty serious developmental disorders in mammals. In plants, they tolerate a lot more genetic change. We've talked about how they can be polyploid, octoploid, things like that, but they also see these kinds of wholesale changes to chromosome structure or number. One of the ways to get abnormal chromosomal number is through a process called non-disjunction. So non-disjunction is basically a mistake that occurs when homologous chromosomes don't separate during meiosis, or when sister chromatids don't separate normally during meiosis. In the case of homologous chromosomes, one gamete is going to receive 2 of the same type of chromosome, and the other gamete receives no copies. So you can see this here. A non-disjunction event in meiosis 1 is going to result in 2 gametes that have 1 extra chromosome, 2 gametes that have 1 lost chromosome. If you do this in meiosis 2, a non-disjunction event there, you're going to have 1 gamete with an extra chromosome, 1 without, and 2 that are normal. But in any case, getting any sorts of gametes that have the wrong number of chromosomes is going to create a pretty substantial problem for the zygote and the offspring. Aneuploidy is what happens when those gametes that have the wrong number of chromosomes fertilize. So offspring that have aneuploidy have an abnormal number of a particular chromosome. So a monosomic zygote would only have 1 copy of a particular chromosome. A trisomic would have 3 copies of a particular chromosome, and that's going to depend whether you combine with a gamete that has an extra or a gamete that's missing 1. Polyploidy is if the entire set of chromosomes is multiplied. So triploidy would be getting 3 full sets of chromosomes. Tetraploidy would be 4 full sets of chromosomes. This is common in plants and not in animals, and actually polyploidy is actually a more normal phenotype appearance-wise than aneuploidy, because basically you're doubling everything, you're not missing anything in particular. You can also have some alterations to chromosome structure. So instead of thinking about an entire chromosome missing, you can have some events happen during meiosis that basically break or change the chromosome structure. You can delete a portion, you can duplicate a portion, you can invert, you can cut it off, flip it back, and stick it back on, or you can translocate it. You can move it from one chromosome to another. Here's deletion, so this D portion and whatever genes are found on that large portion D are now gone. Duplication, whatever genes are on B and C, you now have 2 of them, 2 copies of them. Inversion, B, C, D flips, so now it goes A, D, C, B. Inversions aren't necessarily the biggest of these problems because everything still exists, but it might change the expression of different things. And in translocation, you take a portion from one chromosome and attach it to the end of another chromosome. So AB from here is going to go over here to PQR, MNO is going to come over here. And again, this might not be a huge problem, except it's probably going to change expression of different alleles inside of the organism. So diploids, that's homozygous for a large deletion. So if you're missing a lot of chromosome, you're probably missing a lot of genes. A lot of them are essential, and that condition is generally going to be lethal. So if you're missing parts of an entire chromosome, or you're homozygous for loss of entire parts of chromosomes, that's going to be lethal. Duplications and translocations tend to be harmful as well, and like I said, inversions are sometimes better, but it's still going to affect phenotype, and it's still going to influence expression. You don't want any of these things to be happening inside of a cell. So the final lap here, just to kind of go through some human disorders. You've probably heard of a lot of these human disorders due to chromosomal alterations, but it's helpful for you to just hear them and see them and kind of know what they are. There are some serious ones that are caused by alterations of chromosomal numbers. There are some that are aneuploidy that upset the balance more than others, and some of these individuals do survive beyond, and they have a set of symptoms or a syndrome characteristic of the type of aneuploidy. And the reason we have symptoms or syndromes is because when we're talking about missing or changing a large portion of the chromosome, it typically involves a bunch of genes. So we're not talking about a singular genetic event here, we're talking about a pretty substantial genetic event that's occurring. One of the most common ones that you've probably heard of is Down syndrome. That's aneuploidy caused by 3 copies of chromosome 21. It affects about 1 out of every 830 children born in the U.S. And that frequency of Down syndrome increases with the age of the mother. Correlation that has not been explained probably specifically, but you can imagine that meiosis becomes more challenging the older a mother gets. So the likelihood of having a meiotic event that creates a non-disjunction increases with the age of the mother. You can see here is a karyotype of a person with Down syndrome, and if you see the 21st chromosome, there are 3 copies of that chromosome. You can also have aneuploidy of sex chromosomes. These become somewhat interesting. You can have something called Klinefelter syndrome, XXY, so a male with an extra X chromosome. You can have 1 in every 1,000 males born with an extra Y chromosome, and that doesn't, as far as we know, have any defined syndrome. You can have a female with a trisomy, actually 3 X's, XXX. They're slightly taller than average, but they don't really have unusual features. And a lot of these, we don't know what they do because there's not a good reason to kind of track the entire population of who has these aneuploidies if that's not actually creating any sort of medical conditions. And there's also a monosomy on X, which creates females with 1 X chromosome, and they put an O there to recognize they only have 1 X. This is the only viable monosomy in humans, so the only human chromosome that you can only have 1 copy of. XO females are sterile, but otherwise I don't think major medical issues. You can also have some disorders with structurally altered. You've probably heard of cry du cat, cry of the cat. That's a deletion in chromosome 5. Severe intellectual disability, a cat-like cry at birth, and usually die very early. Because if you think of a huge part of chromosome 5 missing, there's a lot of genes important there. And there are some cancers, most notably CML, chronic myelogenous leukemia, caused by translocation of a chromosome. So here is chromosome 9, chromosome 22, you translocate portions like that. This is called the Philadelphia chromosome, just based off its discovery and what it does to cause CML. So the last portion there just kind of flew through some different human traits caused by chromosomes. There's a lot of stuff going on in this chapter, and we will go over all of it in class, but hopefully this helped you see some of the complexity that we can get in chromosomal movement, chromosomal arrangement, and how that can add more diversity to populations that are having offspring. Alright guys, thanks.